students. Uh, Today in this video I'll discuss about Human Genome Project, which is also known as HGP. So first of all, it was conceived in 1984, but officially it began in earliest October 1990. So it was a large multi-centric international collaboration of some companies whose aim was to determine the nucleotide sequence of the entire nuclear genome of a human. So in 1997, US established NHGRI, which means National Human Genome Research Institute, and HEP was international research group from 6 countries, and the 6 countries involved were USA, UK, France, Germany, Japan, and China. And several laboratories and large number of scientists were involved here in this discipline. The collaboration venture was named as IHGHC, International Human Genome Sequencing Cons- Consortium, right? So the main objective earliest in the history of the project was to obtain the complete sequence of the DNA, and that was extracted from the cells, which was donated by some donors, so that a sequence of DNA can be determined in each chromosomes, in haploid chromosomes of human beings. And also the aim was to construct the genetic map to facilitate the linkage of gene studies, also to discover all the human genes to allow the further studies of human genetic diseases, and to develop this simplified and automated technology for DNA sequencing process. Now, HEP result was attracted worldwide attention, and the achievement was hailed with many description in the media. The mystery of life unraveled, the library of life, periodic table of the life, holy grail of human genetics, and the total number of genes in the human genome is in the range of 33,000 to 44,000. Around 75% of these genes have the same DNA sequence in all the individuals except for those with rare mutations. Any two human genomes are approximately 99.9% identical. Only 0.1% has significantly changed in personality, behavior, in their intelligence, disease, susceptibility, or maybe other type of traits. So what was the goal of HEP? To establish the complete human genome sequence so that it can be freely accessible, to improve the sequencing technology by developing new and more effective methods to analyze the sequence variation in human genome, such as single nucleotide polymorphisms, SNPs, and other DNA sequences variations. Also to develop the technology for functional genomics, and it includes development of additional cDNA resources and technology to analyze the detailed genes expression and the comprehensive study of function of known protein coding sequence, and encourage the development of technology for global protein analysis. Also to study the comparative genomics by completing the genome sequence of some model organisms, and the model organism was taken at that time was mouse, which enhanced the understanding of the human genome. So they also consider the ethical, legal, and social implication ताकि आगे problem ना आए कोई, and anticipated the clash of these new and advanced technology with pre-existing ethical philosophical perspectives, which may be undesirable consequences, right? So what was the medical implication at that time? So it was a huge amount of genetic information obtained from HEP. It serves as a resource for identification of human disease. For example, in oncology, if we see the sequence changes in the cancer cells, that can be identified easily by looking at the genome of the sequence against the draft genome. So advancement of biotechnology with regard to development of useful genes also expand the scope of gene therapy and open the new ways of combating the diseases. Also, it initiate a new field such as pharmacogenomics, which would individualize the therapies depending on the genetic makeup of any type of a patients. Now, what is the application of HGP? To identify the human genome and their function, understand the polygenic disorders, which actually pass on from the genes, and also to improve the gene therapy and diagnosis of the disease. It also developed the pharmacogenesis, right? So this is all about HG